That's weird. May change this format. Okay, can somebody please confirm you can hear me?
I can hear you.
Thank you. I think I cleared the Tuesday, Thursday, and I meant to clear. I think I cleared the Monday, Wednesday, and I meant to clear the Tuesday, Thursday. I think this is the slide we had covered last time. And this is the Tuesday, Thursday. All right. Are there any questions before we begin? If not, let's take a look at the schedule. Understand that the schedule is a day off, meaning a class day off, and I'll need to update that. I'll try and update that this Friday. So today's the January 24th, and we're supposed to be starting chapter 3 and talking about chapter 3. In reality, we're a day behind, so we're still on chapter 2. Let me remind you, you do have a quiz today. It needs to be finished by 11:59 p.m. And this quiz covers chapter 1, chapter 10, the start of chapter 2, what we covered on Monday, and then labs 0, 1, and 2. And all of those labs have been graded for the first time. Any question about any of that? All right. So let's continue with chapter 2. So we talked about the simple lipids and said that triglycerides are made by combining 3 fatty acids with 1 glycerol. Let's move on to the phospholipids. The phospholipids are the main molecule in the cell membrane, at least of eukaryotes and bacteria. A phospholipid is an unusual lipid because it is, well, it is got a polar region, so it can dissolve in water. So it is not hydrophobic the way most lipids, actually, I think all other lipids are. Phospholipid is shown here. Let me blow this up. And it's composed of 2 fatty acids connected to a glycerol. And on the 3rd position of glycerol, we have a phosphate group. This phosphate group is polar, so is the glycerol slightly. And this makes the phospholipid polar on this end. We call this end the head. that has the phosphate group. And then the other end is called the tails, which have the fatty acid group, and they are non-polar. This region is hydrophobic. This region is hydrophilic. This makes this molecule biphasic. There are not too many molecules which are biphasic, but you do know or you're familiar with one other, and that is soaps and detergents. And the reason why soap and detergents clean better than water alone is one end of the soap and detergent can get in and dissolve in the oils, and that will clean out the oils from our clothes. And the other end is polar, and that end can then dissolve in water. And so the detergents can both dissolve in water and in oil, and that's sort of the same as a phospholipid. One end can dissolve in water, the other end can dissolve in oil, making this molecule biphasic. Any question about any of that? So we call this end with the phosphate region the head, and it is polar. I didn't mention the phosphate is also attached to an R group. That's an organic molecule. R can stand for essentially anything, but all you need to know is there is an organic molecule um, attached to the phosphate. It's the phosphate which mainly makes the phospholipid have a polar end. And then the saturated and unsaturated fatty acids make the phospholipid have a non-polar, what's the word I'm looking for? Hydrophobic end. So this is the molecule. And when we put them together, especially in water, they'll start lining up like this, where the tails, which are hydrophobic, will come together, and that way they'll be away from water. And then the heads, which are hydrophilic, will face the water. And in water, this will come around on itself, forming this structure and maybe a little bit of this structure, but I'm not going to talk about a micelle. I'm only going to talk about the liposome. So in this liposome, we have the phospholipid arranged where in a bilayer with the fatty acid tails on the inside, and then the polar phosphate group on the outside, and then the polar phosphate group on the inside. And there can be water in here and obviously water out here. What does that remind you of? What does that even look like? Come on, somebody got a guess?
It looks like a cell.
Yes, it looks like a cell. And that is exactly kind of what a cell is. And that is it's mostly water on the inside. Cytoplasm is mostly water. And then on the outside of a cell, there's also mostly water. You can think of that as a blood cell, a red blood cell in the blood, and the blood is obviously mostly water. In reality, that can apply to any cell because the human body or any organism is mostly water. So outside the cell is mostly water. Any question about that? And in the cell, we have the phosphate of the phospholipid facing outside the cell, facing the water, and facing inside the cell, facing the water of the cytoplasm. And then the phospholipid fatty acid tails are all on the inside, away from the water. And that is exactly how a cell membrane is. Any question about that? No, I guess I should let's back up one. I guess I should mention that the cell membrane does have other molecules in it. It can have proteins in it, and it can have lipids in it, and it can have carbohydrates in it. But phospholipid is by far the most common molecule in the cell membrane, or any membrane for that matter. And it is the phospholipid bilayer that gives the cell membrane its properties, meaning it's the phospholipid bilayer that allows certain molecules to enter the cell and other molecules to leave the cell. And it does not allow other molecules to enter or leave the cell unless there's a transport protein to transport the molecule across the cell membrane. Any questions about any of that? All right. Let's talk about the last group of lipids that we're going to talk about, and that is the steroids. The steroids, unlike the other lipids, are not composed of monomers. For example, the monomers of phospholipid are the phosphate portion, the glycerol portion, and then the fatty acid portion. The triglycerides are monomers of fatty acids and glycerol. But not all lipids are polymers made up of monomers. And the steroids and the sterols are one. It is true there is another group of lipids that we're not going to talk about, and I don't even think your textbook author discusses them. But another lipid is a wax, and it is like a steroid in that it's not made up of monomers. A steroid is a 4 interconnected rigid carbon ring characterizing the group. The most famous is cholesterol, shown here, and it has 4 rigid carbon rings. If there's an OH group attached to the steroid, we call it a sterol. So cholesterol, which has the ol, is a sterol. Sterols, particularly cholesterol, is an important part of the animal cell membrane. So here we have our cell membrane in an animal cell that is mainly phospholipids, but it does have cholesterol in the cell membrane. And the cholesterol gives the cell membrane strength. It's thought that this is because the animal cells do not have a cell wall to protect the cell membrane. And so animals put cholesterol in their cell membrane to help strengthen the cell membrane. Cholesterol is only found in animals. You do not find it in plants. And as you know, a plant cell has a cell wall which will protect the cell membrane. Any question about any of that? All right. Are there any question about the lipids? If not, let's move on to the proteins, the third biological molecule we want to talk about. Proteins are essential in cell structure and in cell function. Proteins may do a diverse number of things or functions. There can be structural proteins that give structure to something, and these are found in bone, muscle, and hair. Proteins can be enzymes, and these molecules speed up chemical reactions. Proteins can also be transport proteins that move chemicals across the cell membrane or across a organelle membrane if we're talking about eukaryotic cells. Proteins can also be regulatory proteins that regulate a body function, such as a hormone and a growth hormone is a protein that children, for example, make to help them grow. There are proteins responsible for movement, both in our muscles that move, and why we can walk and move our arms is because of proteins, as well as, and we're not going to talk about our muscle proteins, but we will talk about proteins in cells that can move. And there are proteins in a flagella, and it's the proteins that make the flagella move, making the cell move. There are proteins responsible for some bacterial toxins. So some proteins can be a bacterial toxin. If you don't know, a toxin is a substance which is toxic to us. And then there are some proteins that we call antibodies that are made by organisms to help defend the organism against a microbial infection. So they help defend the body. Any question about proteins? Well, hopefully you've seen that proteins are very diverse and they have a large number of different functions in an organism. Why proteins are more diverse than any of the other biological molecule is mainly because proteins have more monomers, meaning proteins are made up of more monomers than any other of the biological molecules. You have a handful of different monomers and carbohydrates, and you have a handful of different monomers and lipids as well. There's only 4 monomers in DNA and RNA, and we'll talk about those next. But in proteins, at least in humans, we have 20 different monomers which make up the protein. And most organisms have 20 different monomers that make up a protein. We call these monomers that make up the protein amino acids. Amino acid is shown here. It has a central carbon with hydrogen attached to that central carbon. On one end of the carbon is an amino group, NH2. On the other end of the carbon, opposite the amino group, is the carboxyl group, COOH. And of course, that first O has a double covalent bond to the carbon. There's also an R group on the amino acid. And as I mentioned earlier, R can stand for essentially anything. It's usually an organic molecule, but I guess in some cases it's actually an inorganic molecule. And there are 20 different R groups because it's the R group that determines which amino acid it is. So there's one R group for each different amino acid. And there's one R group shown here for the amino acid tyrosine. Before I go on, I should mention that the amino acids are linked together one after the other to form proteins. Coming back to the amino acids I mentioned in humans, there are 20 different amino acids. And it is true that in most organisms, they have 20 different amino acids. So most bacteria also have 20 amino acids, but they may have different amino acids than we do. But usually when they throw out one, they only add one. I think the highest amino acids ever found in a living microbe was 23. And you don't need to know that, I'm just stating it. You do not need to know these examples of the amino acids. I'm just trying to show you them. So don't memorize them. The simplest amino acid having the simplest R group is glycine, which simply has the R group, a single hydrogen atom attached. Come on, mouse. For some reason, my mouse died. There it is. Attached to the central carbon. You can have a non-polar group like methane attached to the central carbon, and that's what happens in alanine. You can have branched methanes in valine and longer branched in leucine or isoleucine. I guess I should back up. All of these including that one, all of these R groups are nonpolar. That isn't always the case. Here's a few other R groups. This R group is nonpolar, but this group is polar right here on that OH group. The rest of the molecule is nonpolar, but that portion of the R group is polar. This portion and that portion is polar. The rest of the R group is nonpolar. That's mainly this region of the molecule here. And then this one is mostly nonpolar, but it is polar at this NH group. The important thing I'm trying to show you is that the R group can be nonpolar, like this one here, phenylalanine, nonpolar R group. But some of the R groups can be polar. And then the amino acids always have polar regions, meaning the amino group of every amino acid and the carboxyl group of every amino acid are polar. So proteins can be both, in fact, they are both polar and nonpolar. Certain of the R groups are nonpolar. And then obviously the amino groups and the carboxyl group, and some R groups are polar. Any question about any of that? All right, so amino acids are linked together by a peptide bond. A peptide bond is where a nitrogen is bound to a carbon. That's actually a rare bond, not only in living organisms, but it's a rare bond in the universe. Nitrogen isn't bound too often to carbon. It is common, though, in proteins, and that's why we call it a peptide bond, because a peptide is a growing protein. And when you're first making the protein, you attach two amino acids together like this. OH comes off of one amino acid, the H comes off of the other amino acid, and then that nitrogen binds directly to the carbon. and water is formed. A peptide can be 2 amino acids up until several amino acids. I'm not sure exactly what the upper limit is. And then there's a polypeptide, which is many amino acids linked together. And the polypeptide is the growing protein all the way up until you add the last amino acid. So we call that growing protein a polypeptide. But once you've added the last amino acid, it's no longer called the polypeptide. Here it's called a protein. And you'll notice I did use protein, and then I used growing protein as a protein that's still getting longer, and that growing protein is a polypeptide, unless it's extremely small, just a few amino acids, in which case it's not a polypeptide, it's a peptide. Hopefully I didn't confuse anyone there. All right, let's move on. Proteins have 4 levels of their structure. There's a primary level, a secondary level, a tertiary level, and a quaternary level. Let's watch a little movie discussing the 4 levels of protein structure. Taking its time.
I'm Aubrey. I went to St. Jude because I had a cancerous.
That's a commercial.
In this video, we're going to talk about the structure of proteins. And what basically is a protein? A protein is a polymer consistent of many amino acids. So each amino acid represented by the circle is a monomer that forms the protein. Whenever you have many amino acids, it's also called a polypeptide. And the bond that connects each individual amino acid residue is a peptide bond. So let's talk about the individual structure of an amino acid. So an amino acid has a chiral carbon with a hydrogen attached to it, and it has an amine group attached to it as well. And then it has an R group and a carboxyl group. Now I'm going to draw another amino acid right next to it. The left side, where the nitrogen is located, is the N-terminal of the amino acid. The right side, that has the carboxyl group, is the C-terminal of the amino acid. Now, these 2 amino acids can react with each other in a condensation reaction where we're going to lose water, and we're going to connect the 2 amino acids into one molecule. So once water is removed, we could form a peptide bond. This is also called a dehydration reaction because you're losing water. And so this is going to be the new product of the reaction. So let's see if I can fit it in here. So this oxygen is gone. So now we have a nitrogen. Let me take out H2O. We have 1 hydrogen left over. We lost 2 once we lost water. So this is a dipeptide because we have 2 amino acids combined. And so this amide bond that we see here, that is the peptide bond. We formed a bond between the carbon and the nitrogen atom. So whenever you combine two amino acids to form a peptide bond, you're forming a covalent bond, which is hard to break. Now let's talk about the different levels of protein structure. There's 4 levels that you need to know: the primary structure, the secondary structure, the tertiary structure, and finally the quaternary structure. Now the primary structure is simply based on the sequence of the amino acids found in the protein. The sequence determines the shape of the protein. If you replace just one amino acid with another, it will completely change the shape of the protein. So the shape and its function is primarily determined by the sequence of amino acids. The secondary structure describes the localized shape of a protein, and there's 2 of them that you need to be familiar with: the alpha helix and the beta pleated sheet. In the alpha helix, you could see that it's stabilized by hydrogen bonds. The NH group of one amino acid interacts with the carbonyl group of another amino acid, and each turn contains about 3.6 amino acid residues. Now here is a visual illustration of the beta pleated sheet. Just like the alpha helix, it too is stabilized by hydrogen bonds between the carbonyl group of one amino acid and the NH group of another amino acid. Next up, we have the tertiary structure, which represents the 3-dimensional complete folding pattern of the protein. And so here's a visual illustration of it. So we could see some areas is just a straight chain. Here we have an alpha helix, and here we have a beta pleated sheet. Now, the tertiary structure is one individual subunit. When you combine multiple subunits, you create a quaternary structure. So hemoglobin is an example of that. It has 4 individual subunits, 2 alpha subunits and 2 beta subunits. So hemoglobin is a protein with a quaternary structure. And so that's basically it for this video. Hopefully it gave you a basic understanding of proteins and the 4 levels of structure that they have. Thanks for watching.
Okay, any questions on that? So there are 4 levels of protein structure. The primary level is just the specific sequence of amino acids in a polypeptide chain, or really in the protein. So when the protein is first made, it has a primary structure, just the sequence of amino acids. And that protein is also fairly straight when it's first made. The protein then forms the secondary structure. And the secondary structure forms by the interaction of nearby amino acids. So the neighboring amino acids start interacting with each other, and that gives rise to the secondary structure. The number of interactions can be both attraction and repulsion in the secondary structure. And oftentimes, websites on the internet, and maybe your book as well, say that the main interaction for a secondary structure is only hydrogen bonds. And they say that the secondary structure happens because of hydrogen bonds. That isn't correct. And that is, there are many forces of attraction and a few forces of repulsion that give rise to the secondary structure. And it is true, hydrogen bond is one of the major ones, but it is not the only force of attraction. Do I have this blown up? Nope, there we go. So the neighboring amino acids can be attracted to each other or repulsed from each other. And that's what I mean by the neighboring amino acids start interacting with each other. And once they interact with each other, they will give the secondary structure. There are actually three. The video you just watched only discussed two. There's the alpha helix, shown here, the beta pleated sheet, shown here, and I gotta open up Paint to show you the third, which is the random coil. The random coil is just where the protein takes on a random shape. We'll make it like that. So that's an amino acid chain making the polypeptide protein. And actually, this random coil is beyond the secondary stage. This would be the tertiary stage. Anyways, the random coil is something like a shape like that, and it can be any type of a shape. But the point is there be regions which are interacting and then regions which are not interacting. And this is really more the tertiary structure. But oh, where am I? Here we go. So there are three different types of shapes the protein can have from the neighboring amino acids interacting with each other. And these interactions can be attraction, such as a hydrogen bond forming between this amino acid and that amino acid. But it can also be an ionic bond if this amino acid has a positive charge and that amino acid has a negative charge. That will be an ionic attraction between them, and they will be brought closer together, attracted to each other, the same as the hydrogen bond. Another attraction, maybe I'll put it this way because it's not showing a hydrogen bond or an ionic bond. Another attraction is van der Waals forces, which we're not really going to talk about much, and I'm not even sure your textbook talks about it, but that is the attraction of non-polar molecules towards other non-polar molecules. It is a weak attraction, but if you think about it, when you have your bottle of olive oil or corn oil, the reason why it's a liquid is because the molecules of oil are attracted to each other and they don't form hydrogen bonds. It's van der Waals forces where nonpolar molecules are attracted to other nonpolar molecules. And you know it has to be an attraction between the molecules because it's a liquid. If it wasn't an attraction between the molecules, then it wouldn't be a liquid, it would be a gas. And how you can prove that is you can take your olive oil and stick it in the refrigerator. You will lower the temperature where the energy or the temperature will be cool enough that the molecules cannot break the force of attraction between them. And if you don't break the force of attraction between molecules that are attracted to each other, you will not have a liquid. If you cannot break the force of attraction, you will have a solid. and you actually should know that from ice. What happens is instead of the molecules breaking that hydrogen bond between the different water molecules, the temperature of the water is cool enough that that hydrogen bond is never broken. And instead of having liquid water, we have solid water. And the same thing happens with olive oil. You put it in the refrigerator, you will lower the temperature enough so that the force of attraction between the different molecules of oil cannot break. And you no longer will have a liquid. You will have solid olive oil. Any question about any of that? All right, so there can be that interaction or attraction, and that's between two non-polar amino acids. You can also have repulsion. This right here, come on, mouse, can be a non-polar amino acid, and this would be a polar amino acid. These two will then be repulsed from each other. You can also have repulsion where the amino acids have the same charge, meaning if one amino acid has a positive charge and another amino acid also has a positive charge, which in this case, because it's the same amino acid, it'll obviously be the positive charge. And these two amino acids will be repulsed from each other. So if they both had a positive charge, they would be repulsed from each other because positive charge does not like being near another positive charge. Any question about any of that? So those are the different forces of attraction and repulsion that can happen in the secondary structure of a protein. Any question about any of that? All right, once the secondary structure forms, the protein can then supercoil, taking on its close to its three-dimensional shape. And if it's only one polypeptide protein, that will be its three-dimensional final shape. How that happens is the tertiary structure is where the amino acids interact with each other, but it's not just the nearby amino acids which are interacting with each other. It's any amino acid can interact with any other amino acid all over the protein. As shown here, this amino acid, which is at the end of the protein, come on, mouse. This amino acid here, which is at the end of the protein, is being attracted to this amino acid here at the near the start of the protein. And that gives the tertiary structure. These forces of attraction and repulsion between the amino acids of the 3D structure can be all of the forces of attraction and forces of repulsion that we saw in the secondary structure of a protein. However, in the tertiary structure, we have one other force of attraction. Let me see if I can erase this now. I'm not doing too well of a job, but I need a little more room here. Anyways, that's the sulfur dibridge, where on cysteine, here and here, we have the amino acid cysteine. Let me see if I can get that. Yeah, let me see if I can copy that. Got it. Put the cysteine over here. Cysteine has a sulfhydryl group on it. It's not quite right, but maybe I move it up. There it is. So that'll be bound to a sulfhydryl group. And we're going to do it over here as well. Up. There we go. That should be bound a little bit more there. Let me see if I can drop that a little bit. Come on. Yeah. It's apparently too small for me to grab. Now I got it. But now I have to shrink it. A little further. Sort of there. Sorry about that. Anyways, this is the cysteine. It has a sulfhydryl group. Well, the hydrogens can come off of the sulfhydryl group, and they just go into a solution. And once they come off, the hydrogens can then bind, the sulfur can then bind to each other. Oh, I'm just going to do it this way. And that is a covalent bond between the sulfur of the cysteine. This sulfur disulfide bridge is a very strong bond. This is a covalent chemical bond, much stronger than a hydrogen bond. Anyways, that's shown here, where we have the disulfide bridge between the cysteine. This interaction is only seen in the tertiary structure. It's a very strong linkage between this amino acid and that amino acid. Let me think, is that the strongest interaction we've seen between amino acids? Yes, that is. And there can also be, obviously, attraction of hydrogen bonds between amino acids, ionic bonds between amino acids. There can be repulsion between amino acids, which may be maybe this amino acid here. Come on, mouse. This amino acid here can be repulsed from that amino acid here. Why this is going that way and that way. And that repulsion can be anything we saw as repulsion in the secondary structure. They can both be ionic, but the same charge. So they'll be repulsed from each other. They can be one nonpolar, one polar, and that will repulse them. Any protein that only has one polypeptide subunit will only go up to a tertiary structure. Most enzymes only have a tertiary structure, but some proteins have a quaternary structure, and these proteins are made up of more than one polypeptide. The quaternary structure is where the different polypeptides come together and then interact with each other with all of the forces of attraction and the forces of repulsion that we've talked about in the secondary and tertiary levels. You can see all of those in the quaternary level. And this interaction would be between the amino acids of one polypeptide and another polypeptide. So here, we can say this amino acid here is being attracted to this amino acid there. And maybe this amino acid here is being repulsed from this amino acid here. Only proteins that are made up of more than one polypeptide have a quaternary structure. An example of a protein that has a quaternary structure include hemoglobin and the antibodies. Each of these proteins, hemoglobin and antibodies, are actually made up of 4 different polypeptide units. Any question about the levels of protein structure? All right, let's talk about proteins in a little more detail. Proteins can have an overall shape, and there are 2 different shapes the protein takes. There can be globular proteins. These proteins are compact and roughly spherical. Hemoglobin is an example of a globular protein. There are also fibrous or thread-like proteins, and these proteins tend to be in structural fibers or structural units, such as collagen or hair. Proteins can be conjugated to other molecules. Combination of amino acid with either organic or inorganic components, we call them conjugated proteins. The protein is named after their non-protein component, like a glycoproteins are a protein bound to a glycogen. And glycogen is simply a short carbohydrate like a sugar, or maybe a couple of sugars. A lipoprotein is a protein bound to a lipid. Anyways, those are conjugated proteins. A protein can denature. When the protein loses its 3-dimensional shape, which can often happen when hydrogen bonds break, but it can also happen when hydrogen bonds form, like when you put a protein, which should normally be at 37 degrees Celsius, that's body temperature. When you put that protein in the refrigerator, the protein can have hydrogen bonds forming that are not normally present or not normally there when the protein is at 37 degrees and functioning. But the point is the protein is losing its three-dimensional shape. When the protein totally loses its three-dimensional shape, the protein will no longer be functional, and we say the protein is denatured. Now, there are different categories of denaturization, meaning they're slightly denatured, and the protein will have less than 100% activity, but it will have some activity. And let's say high activity, because I'm talking about just slightly losing its shape. And then you lose its shape a little further. Its functioning is decreased even further. And then you change it further and the shape is partially still there, but a lot of it's gone. So the function is still present, but a lot of the function of the protein is gone. And then when the protein is totally denatured, the protein will not be able to function. You can get a protein to denature with temperature, pH, or salt. So if the protein is at the wrong temperature, or at the wrong pH, or the wrong salt concentration, the protein will denature. An example of that is egg whites. When you open the egg, you have liquid egg whites, which are actually, I don't know, translucent or transparent. And you put it into your pan, and then you cook the egg. How you do that is you're just raising the temperature, and that will denature the protein of the egg white, changing its appearance from translucent to white in color. And then you change it from a liquid to a solid as well. Any question about any of that? So if the protein is at the wrong temperature, pH, or salt concentration, the protein will lose its three-dimensional shape and will eventually denature, meaning will be non-functional. Any question about any of that? If not, let's talk about the fourth of the biological molecules, nucleic acids. There are two big ones you need to know. And then there's a third monomer of nucleic acids, which we're also going to discuss. So the two big ones are DNA and RNA. And then the third small monomer that we're going to talk, which is a nucleic acid, is ATP. All right, DNA and RNA consist of a monomer that we call a nucleotide. So the subunits of DNA and RNA are nucleotides. Nucleotides are made up of three parts. There's the pentose sugar, and that's right there. Pentose is just a 5-shaped sugar, 5-carbon sugar. 5-carbon sugar. There is a phosphate group attached to the sugar, and then a nitrogen-containing base also attached to the sugar. The nitrogen-containing base can be a purine or a pyrimidine. In DNA, we have 4 nucleotides, and you can learn the names either with the long name, or it's abbreviation. So there's adenine or A, guanine or G, cytosine or C, and thymine or T. And either way you learn it is fine. I don't care. Adenine and guanine are purines, and cytosine and thymine are pyrimidines. How you can easily remember which is which is the word pyrimidine has a Y in it. The word cytosine and thymine also have Y's in them. So all the pyrimidines have a Y. The purines do not have a Y in them. And that's how you can learn the pyrimidines and the purines from each other. Now, the basis of one strand of DNA bind to the second strand of DNA. And that binding happens by hydrogen bonding. A and a T are complementary, so they will hydrogen bond with each other. So A will bind with T and T will bind with A. At least we're talking about DNA. I'll talk about RNA in a minute. C is complementary to G, so C and G can also pair, where the C will be on one strand of DNA, single strand of DNA, and the G will be on the other single strand of DNA in the double strand of DNA. As shown here, the A and the T have two hydrogen bonds between them. As shown here, and this is not as good, but it does show it. As shown here, the C and the G have three hydrogen bonds between them. All right, so in DNA, we have A, G, C, and T. In RNA, we have A, G, C. We do not have T. It is replaced by uracil. And if you remember the pyrimidines in DNA, and you recall that thymine is replaced by uracil, uracil has to be a pyrimidine because it's replacing thymine in RNA. So uracil is a pyrimidine, even though it doesn't have a Y in it. Any question about any of that? All right, DNA has the sugar deoxyribose. It's a double-stranded molecule. In the DNA molecule, the double molecule, in one strand of the DNA is held together by the sugar phosphate backbone, meaning here we have a sugar. Come on, mouse. Here we have a sugar, there's a phosphate, and that binds to a sugar and then another phosphate. So the single strand of DNA is held together by a covalent bond between the sugar and the phosphate. So we call the sugar phosphate the backbone of a single strand of DNA. On the other hand, the bases do not hold the single strand of DNA together. They are on the steps of the double helicase that shapes like a twisting staircase. So the bases form the steps of this twisted staircase. They do not hold a single strand of DNA together. The bases do, on the other hand, hold the two strands of DNA together. And when they hold the strand together, It's only hydrogen bonds holding the two strands of DNA together. Because it's a hydrogen bond, that means this hydrogen bond here can break and reform all the time. And why the DNA is held together, at least at 37 degrees in our body temperature, is because when this hydrogen bond breaks, there are other hydrogen bonds holding the DNA together. And then this hydrogen bond can reform, and maybe that hydrogen bond will break. All right, any questions about DNA? If not, let's talk about RNA. RNA has a similar sugar as DNA. It's not deoxyribose, it is ribose. And the only difference is ribose has an extra OH group. In that deoxyribose, we're missing one OH group and there's just a hydrogen there. RNA is only single-stranded. At least in our cells, our RNA is only single-stranded. And it's actually in any cell, the RNA is only single-stranded. You have to have a virus before you can have the RNA double-stranded. And we'll talk about viruses much later in the term. With RNA, once again, the sugar phosphate backbone holds a single strand of RNA together. And it holds it together by a chemical covalent bond. The bases for RNA are A, G, C, and U. Uracil replaces thymine in RNA. There are three major kinds of RNA. There's transfer RNA, also called tRNA. There's messenger RNA, also called mRNA. And there's ribosomal RNA, also called rRNA. When we're looking at the RNAs, what is the difference between tRNA and messenger RNA? Are they different nucleotides? That's a question. Does anyone want to say a yes or no? Nobody's going to comment. There's no difference in the RNA of tRNA, messenger RNA, and rRNA, except for the sequence of the RNA. So it's the sequence of the nucleotides that determine whether the RNA is tRNA, messenger RNA, or rRNA. Any question about any of that? So otherwise, tRNA, messenger RNA, and rRNA are the same. The only difference is the sequence of the nucleotides. Any question about any of that? All right. If not, trying to remember, I think there's one other thing I was supposed to say on RNA and skip my mind. Oh no, I don't want to say that. That'll talk about later, and that is when the gene is being transcribed, how does the polymerase know to make tRNA, messenger RNA, or rRNA? And the polymerase doesn't know. It just makes it, and how it makes it is it simply reads the code on the DNA, and the code of the DNA will determine whether tRNA is made, messenger RNA, or rRNA is made. And I just stated it, but you don't need to know that. So let's move on. ATP, adenosine triphosphate, is the last of the members of the nucleic acids I want to talk about. We need to talk about ATP a little bit because it is the primary energy source for cell activities. ATP is used whenever the cell needs energy. It tends to get that energy from ATP. ATP does have ribose. It does have adenine as the nucleotide base. I mean the nitrogen containing base. And then it has 3 phosphate groups attached to it. The third phosphate group has a squiggly line because it is bound to the second phosphate with a high energy bond. And if the cell wants a lot of energy, it just needs to break that third phosphate bond, and that will separate this phosphate from the remainder of the ATP. It's not an ATP anymore, but it's actually ADP, but we'll just say the remainder of the ATP. If the cell needs more energy, it can break off the second phosphate group because it's bound to the first phosphate group with another high energy bond. So ATP is made by dehydration synthesis. To make ATP, you take ADP, meaning adenine with 2 phosphates, you bring on a third phosphate group, and it takes energy. Because you're making a bigger molecule, you need energy coming into the system. And then that will drive the reaction this way, generating water. And then this phosphate binds to that phosphate to make ATP. On the other hand, ATP can be broken down, usually for the purpose of releasing the energy in the high energy third phosphate group. I guess I should be here. Sorry. And how you do that is you take ATP plus water going this way, and that'll make ADP, adenosine diphosphate. And then you get the phosphate group coming off, and the I just stands for an inorganic phosphate group. It has no organic group bound to it. And then energy will be released. The cell usually will do this to release the energy and then use that energy for the cell's energy needs, such as when the cell is making a carbohydrate like starch or a protein such as catalase. All right, any question about ATP? Not, let me go to my last slide. And we've talked about hydrogen bonds in this lesson, as well as in our last lesson. What molecules did we discuss that have hydrogen bonds that give the molecule important properties? So what molecules have we discussed?
H2O.
Yeah, water has hydrogen bonds, and it gives water important properties. Namely, without the hydrogen bonds, you wouldn't have liquid water, because you need to have the hydrogen bonds to form liquid water, as long as the bond can break. Because if it doesn't break, you don't have liquid water, you then have ice. What's another molecule that we've discussed that has hydrogen bonds giving the molecule important properties? Anyone? Well, what of the biological molecules have hydrogen bonds? This is pretty easy, people. What of the biological molecules, there's only 4 of them, has hydrogen bonds?
Proteins.
Can you say that again? You sort of broke up.
Proteins.
Proteins have hydrogen bonds. Actually, nucleic acid and carbohydrates also have hydrogen bonds. Nucleic acids is the one I just wanted to mention because it's very important. Without the hydrogen bonds, you wouldn't get double-stranded DNA. The reason why we have double-stranded DNA is because of the hydrogen bonds forming between one strand with its complementary strand. And then lastly, what would happen to all these molecules, proteins, nucleic acids, and water, if all of these hydrogen bonds were to suddenly disappear?
Would have no way of forming or holding shape.
Yeah, the double strand DNA, if you lose all the hydrogen bonds, you'll have single strand DNA. The protein will likely denature, probably 100%, because there's a lot of hydrogen bonds in a protein. Okay, any questions about anything? If not, I'm going to end it here and I will see you at 6:30 for the lab. Bye.